All right, hello everyone. Well, I'm here to talk about a piece that I recently wrote about biological evolution. It's called Why Does Biological Evolution Work? A Minimal Model for Biological Evolution and Other Adaptive Processes. And we're going to kind of go through the piece that I wrote, and then there hopefully will be time for some questions and answers at the end. So this is the piece I wrote. Yeah. And we're going to start off going through this. So first, we can talk about the model. Why does biological evolution work? And for that matter, why does machine learning work? Both are examples of adaptive processes that surprise us with what they manage to achieve. So what's the essence of what's going on? I'm going to concentrate here on biological evolution, though much of what I'll discuss is also relevant to machine learning, but I'll plan to explore that in more detail elsewhere. Okay, so what is an appropriate minimal model for biology? My core idea here is to think of biological organisms as computational systems that develop by following simple underlying rules. And these underlying rules, in effect, correspond to the genotype of the organism. The result of running them is, in effect, its phenotype. Cellular automata provide a convenient example of this kind of setup. And so let me show you an example here of this involves cells with three possible colors. So the rules are shown over on the left, and the behavior they generate is shown on the right. So we're starting from a single kind of orange cell here. We see that from this seed, a structure is grown, which in this case dies out after 51 steps. In a sense, it's already remarkable that we can generate a structure that neither goes on forever nor dies out quickly, but instead manages to live, in this case, for exactly 51 steps. But let's say that we start from the trivial kind of null rule that makes any pattern die out immediately. Can we end up adaptively evolving to the rule that we see here? Imagine making a sequence of randomly chosen point mutations, each changing just one outcome in the rule like this. Then suppose that at each step in a minimal analog of natural selection, we accept any mutation that makes the lifetime longer, though not infinite, or at least the same as before, and we reject any mutation that makes the lifetime shorter or infinite. It turns out that with this procedure, we can indeed adaptively evolve to the rule that we showed before. And here we're just showing kind of the waypoints of progressively greater lifetimes. So different sequences of random mutations give different random sequences of rules, but the remarkable fact is that in almost all cases, it's possible to make progress and routinely reach rules that give long-lived patterns. So let me show you examples of that. So here's an example with a lifetime of 100 that reaches eventually lifetimes of 107 and 723 steps. Here's one that reaches lifetime of 723 steps, always producing kind of elaborate morphological structure. Is it obvious that our simple process of adaptive evolution will be able to successfully kind of wrangle things to achieve something like this, living 700 and something steps? No, it's not obvious. But the fact that it can do this seems to be at the heart of why biological evolution manages to work. So looking at the sequences of pictures like the one we see here, we see that there are often, in effect, different mechanisms for producing long lifetimes that emerge in different sequences of rules. Typically, we first see the mechanism in kind of a simple form, like at the top here. Then as the adaptive process continues, the mechanism gets progressively more developed, elaborated, and built on, not unlike what we often appear to see in the fossil record of biological evolution. But let's drill down and look in a little bit more detail at what's happening in the simple model we're using. In the three-color nearest neighbor, that's k equals 3, r equals 1, cellular automata, we're considering there are 26, that's 3 cubed minus 1, relevant cases in the rule. There'd be 27 if we didn't insist that the all white sequence of rules goes to all a white cell. So point mutations affect a single case in the rule, changing it to one of two, i.e. 3 minus 1, possible alternative outcomes. So there are altogether 52, which is 26 times 2, possible distinct point mutations that can be made to a given rule. So for example, if we start from this rule, the result of possible single point mutations are these. And that's a sequence that goes on. And even with such point mutations, there's usually considerable diversity in the behavior they generate. So here are the results for all of the point mutations in this case. In quite a few cases, the pattern generated is exactly the same as the one for the original rule. In other cases, it dies out more quickly or doesn't die out at all, either becoming periodic or growing forever. And in this particular example, in just one case, it achieves higher thickness, surviving longer. So that's that case right there. So if we make a sequence of random mutations, many will produce shorter-lived or infinite lifetime kind of tumor patterns, and these will reject, or in biological terms, we can imagine they're selected out. But there is still plenty of there can still be many kind of neutral mutations that don't change the final pattern, or at least give a pattern of the same length. And at first, we might think that these don't achieve anything, but actually they're critical in allowing single point mutations to build up to larger mutations that can eventually give long-lived patterns. So that's a sequence of those mutations kind of at first not making a change to the phenotype, the pattern that's generated, but changing the genotype and eventually changing the phenotype as well. So tracing our whole adaptive evolution process, like the one we saw at the beginning here, the total number of point mutations involved in getting from one increasingly long-lived fitness waypoint to another is what we see here. So, and we can then look at the kind of underlying rules associated with these fitness waypoints. And the numbers here just count the number of cumulative accepted mutations, ignoring ones that kind of just go back and forth. So one can get a sense of what's going on with the whole sort of sequence of accepted mutations, accepted rules in this adaptive evolution by plotting them in a kind of dimension-reduced rendering of this 27-dimensional rule space. So here's a plot of that. And there are periods when there's sort of a lot of just wandering around going on, with many mutations needed to make any progress. And then there are other periods when things go much faster and fewer mutations are needed. Well, as another way to see what's going on, we can plot kind of the maximum lifetime achieved so far against the total number of mutation steps made. So we see lots of plateaus, including an extremely long one, in which no progress is made, punctuated by sometimes quite large, sudden changes, often brought on by just a single mutation. So if we include rejected mutations, we can see there's lots of activity going on, even in plateaus. Every red dot is the result of a mutation. It doesn't just manage to make progress. In a sense, we can think of any red dot that lies below a plateau as being like a mutation, or for that matter, an organism that doesn't make it and is selected out. So it's worth noting that there can be multiple different kind of phenotype patterns that occur across one of these plateaus. Here's what we see in this particular example. So, and what we're seeing there is in these different blocks are the different phenotypes that occur across equal fitness plateaus. But even between these phenotypically different cases, there can be many genotypically different rules. In a sense, this isn't surprising because usually only parts of the underlying rule are coding. Other parts are non-coding in the sense that they're not sampled during the generation of the pattern from that rule. And for example, here's a picture that highlights go. For each fitness waypoint rule, which cells make use of a fresh case in the rule that hasn't already been so far sampled during the generation of the pattern? So every kind of highlighted cell here is a cell that makes use of a new case in the underlying rule that generates the final pattern. So we see that even in the last rule shown here, only 18 of the 26 relevant cases in the rule are actually ever sampled during the generation of the pattern, at least from the particular single red cell initial condition that's used. So this means that 8 cases in the rule are undetermined from the phenotype, implying that there are 3 to the 8 or 6,561 possible genotypes, i.e. rules, that will give the same result as that final result here. Okay, well, so far we've mostly been talking about one particular random sequence of mutations, but what happens if we look at many possible such sequences? So here's how the longest lifetime, or in effect fitness, increases for 100 different sequences of random mutations. And what's perhaps most notable here is that it seems as if these adaptive processes indeed don't get stuck. It may take a while, as the result that there are long plateaus, but these pictures suggest that eventually adaptive evolution will find a way, and one will get to rules that show longer lifetimes. So we can kind of see another reflection of that if we look at the progressive development of the distribution of lifetimes as a function of the number of adaptive evolution steps. We see that we get this histogram, which is heading to the right to longer lifetimes and away from the left, which corresponds to short lifetimes. Okay, let's go on. Let's talk about the multi-way graph of all possible mutation histories. So in what we've done so far, we've always been talking about particular paths of adaptive evolution determined by particular sequences of random mutations. But a powerful way to get a more global view of the process of adaptive evolution is to look, in the spirit of our physics project, the Ruliad, and so on, not just at individual paths of adaptive evolution, but instead at the multi-way graph of all possible paths. And in making a correspondence with biology, multi-way graphs give us a way to talk about adaptive evolution, not just of individual sequences of organisms, but also populations. So to start our discussion, let's consider not the 3-color cellular automata that we just talked about before, but instead nearest neighbor 2-color cellular automata, for which there are just 128 possible relevant rules. And how are these rules related by point mutations? Well, we can construct a graph of every possible way that one rule from this set can be transformed to another by a single point mutation. Here's that graph. So if we imagine 5-bit rather than these 7-bit rules, there are only 16 possible ones, and we can readily see that the graph of possible mutations has the form of a Boolean hypercube. Well, let's say we start from the null rule. That's the rule which is all white down in the bottom right-hand corner there. Then we enumerate the rules obtained by a single point mutation, and therefore directly connected to the null rule in this graph. We see what behavior they can produce, and say from a particular initial condition. There's the result. So the caption to those evolutions in each case is the sequence of cases, the sequence of bits that represent the rule. Some of these rules we can view as making progress in the sense that they yield patterns with longer lifetimes, not impressively longer, just 2 rather than 1. But other rules make no progress or generate patterns that live forever. So keeping only mutations that don't lead to shorter or infinite lifetimes, we can construct a multi-way graph that shows all possible mutation paths. And here's that graph. So although there's a very small graph with just 15 rules appearing, we can already see hints of some important phenomena. There are kind of fitness neutral mutations that go both ways, but there are also plenty of mutations that go one way, because the other way, they would decrease fitness. And a notable feature of this graph is that once one's committed to a particular part of the graph, say the left or the right, one often can't reach a different one, suggesting an analogy to the existence of different branches in the tree of life. So moving beyond 2-color nearest neighbor, that is k equals 2, r equals 1 cellular automata, we can consider k equals 2, r equals 3 halves ones. So a typical such cellular automaton is this one here. And for k equals 2, r equals 1, there are a total of 128, 2 to the 2 to the 3 minus 1 relevant rules. For k equals 2, r equals 3 halves, there are a total of 32,768, or 2 to the 2 to the 4 minus 1 rules. So starting from the null rule, and again using a particular initial condition, here are a few specific examples of evolution paths that we get for cellular automata of this type. And now we can look at, here's the beginning of the multiway graph for, doesn't look quite right because we can't see some gray outlines, but it's okay. We get the basic point. This is the beginning of the multiway graph for k equals 2, r equals 3 halves rules, showing rules reached by up to 2 mutations, 1 level, 2 levels, from the original null rule. So this graph contains many examples of fitness-neutral sets, rules that have the same fitness and that can be transformed into each other by mutations, back and forth. So a few examples of these fitness-neutral sets are these ones here. And in the first case here, the morphology of the phenotypic patterns is the same for all genotypic rules in the fitness-neutral set. But in the other cases, there are multiple morphologies within a single fitness-neutral set. If we included all individual rules, we could get a complete k equals 2, 3, r equals 3 halves multiway graph with a total of 1,884 nodes. But if we include just one representative from every fitness-neutral set, we get a more manageable multiway graph here with 86 possible nodes. So keeping only one representative from pairs of patterns that are related by left-right symmetry, we get a still simpler graph, now with a total of 49 nodes. And there's quite a lot of structure in this graph, with both divergence and convergence of possible paths. But overall, there's a certain sense that different sections of the graph separate into distinct branches in which adaptive evolution, in effect, pursues different ideas about how to increase fitness, i.e., in this case, lifetime of patterns. So we can think of these fitness-neutral sets as representing, we think of the fitness-neutral sets that appear here as representing a kind of equivalence class of rules. There's quite a range of possible structures to these sets, from ones with a single element to ones with many elements, but few distinct morphologies, to ones with different morphologies for every element. And so here's a representation of all the fitness-neutral sets for k equals 2, r equals 3 halves cellular automata. And the little graphs on the left show the kind of relationship between those elements and the fitness-neutral set, and on the right are the morphologically different examples of phenotypes in that fitness-neutral set. What about larger spaces of rules? For k equals 2, r equals 2, that's nearest neighbors on both sides. They're altogether about 2 billion, or 2 to the 2 to the 5 minus 1 relevant rules. But if we choose to look only at left-right symmetric ones, this number is reduced to 524,288, or 2 to the 19. And let's look at, here are some examples of sequences of rules produced by adaptive evolution, in this particular case of symmetric rules, starting from the null rule and allowing only mutations that preserve symmetry. And now we're, again, just using a single black cell as the initial condition to grow our patterns. So once again, we can identify fitness-neutral sets, though this time in the vast majority of cases, the patterns generated by all members of a given set are morphologically the same. And here are some fitness-neutral sets in this case. If we reduce out the fitness-neutral sets, we can then compute kind of the complete transitively reduced multiway graph for symmetric k equals 2, r equals 2 rules containing a total of 60 nodes. And oops, here's the result. By transitive reduction, I mean that there are also cases where there are mutations that lead directly from a phenotype high on this picture to one lower on the picture, but we're only showing the the kind of the slowest path to make that transition, rather than showing all the possible paths in this particular case. So by reducing out fitness neutral sets, we're creating a multi-way graph in which each edge represents a mutation that makes progress in increasing fitness. But actual paths of adaptive evolution based on random sequences of mutations can do any amount of rattling around within fitness neutral sets, not to mention trying mutations that decrease fitness before reaching mutations that make progress. So this means that even though the reduced multi-way graph we've drawn suggests that the minimum number of steps, i.e. mutations, needed to adaptively evolve from the null rule to any other rule is just 9 steps, it can actually take any number of steps because of the rattling around within fitness neutral sets. So here's an example of a sequence of accepted mutations in a particular adaptive evolution process, with mutations that make progress highlighted and the numbers between indicating rejected mutations. So we can sort of see rattling around in fitness neutral sets with a cycle of morphologies being generated. But because we can see that there are morphologies here that are the same or that kind of cycle around. But while this represents one way to reach the final pattern, there are also plenty of others, potentially involving many fewer mutations. And indeed, one can determine from the multi-way graph that the absolutely shortest path to reach that endpoint is this one here. And this involves this sequence of rules. So we're starting from the null rule here, and at each step, we're making a single point mutation, though because of symmetry, 2 bits can sometimes be changed. The first few mutations don't end up changing the phenotypic behavior. Remember, it looked like this. But after a while, enough mutations, here actually 6, have built up that we get morphologically different behavior. And after just 3 more mutations, we end up with our final pattern. So our original random sequence of mutations gets to the same result, but in a much more tortuous way, doing a total of 169 mutations, which often cancel each other out. So in drawing a multi-way graph, we're defining what evolutionary paths are possible. But what about probabilities? If we assume that every point mutation is equally likely, then we can in effect analyze the flow in the multi-way graph and determine the ultimate probability that each rule will be generated with here higher probabilities for generation shown redder. Actually not so obvious in this projected image that there are some reds are redder than other reds here. All right, well, let's go on. Let's talk about the fitness landscape. So multi-way graphs, like this one here, give a very global view of adaptive evolution. But in understanding the process of adaptive evolution, it's also often useful to think somewhat more locally. We can imagine that all the possible rules are laid out in a certain space, and that adaptive evolution is trying to find appropriate paths in that space. Potentially, we can suppose that there's a fitness landscape defined in the space, and that adaptive evolution is trying to follow a path that progressively ascends to higher peaks of fitness. So let's consider again the very first example that I gave here of adaptive evolution in the space of 3-color cellular automata. At each step, so just a reminder what that looked like, that's this case here. So at each step in this adaptive evolution, there are 52 possible point mutations that can be made to the rule. We can think of each of these mutations as corresponding to making an elementary move in a different direction in the 26-dimensional space of rules. So here's kind of a visual representation of what's going on based on the particular path of adaptive evolution from the example that I just gave. What we're showing here is in effect, the sequence of decisions that are being made to get us from one fitness waypoint to another. So different possible mutations are represented by different radial directions, with the length of each line being proportional to the fitness achieved by doing that mutation. So at each step, there's kind of a gray disk that you can't see very well here, that represents the previous fitness. It's kind of where most of the lines are going out to more or less the position of that gray disk. Many possible mutations lead to lower fitness outcomes, which are effectively within the disk. But there are at least some mutations that have higher fitness and escape the disk. So in the multiway graph, we trace every mutation that leads to higher fitness. But for a particular path of adaptive evolution that we're talking about here, we imagine that we always just pick at random one mutation from this set. That's indicated here by a red line. Later, we'll talk about different strategies for the adaptive evolution process. So these kind of radial icons can be thought of as giving a representation of the local derivative at each point in the space of rules, with longer lines corresponding to directions with larger slopes up the fitness landscape. But what happens if we want to knit together these local derivatives to form a picture of the whole space? Needless to say, it's complicated. As a first example, consider just k equals 2, r equals 1, the elementary cellular automaton rules. There are a total of 128 relevant such rules that can be thought of as connected by point mutations that form a 7-dimensional Boolean hypercube. And we already talked about the fact that of 128 relevant rules, only 15 appear in adaptive evolution processes. The others are, in effect, never selected because they represent lower fitness. But now we can ask where the rules that do appear lie on the whole hypercube. And this is a picture of that. So each node here represents a rule with the size of the highlighted nodes indicating their corresponding fitness, computed from lifetime with a particular initial condition. So the node that's shown in green corresponds to the null rule. So rendering this in 3D, we get, and showing fitness as height, as well as size of sphere, we get what we can consider a fitness landscape. And now we can think of our adaptive evolution as proceeding along paths that never go to nodes with lower height on this landscape. So if we look at k equals 2, r equals 3 halves rules, we get a more filled-in landscape. This is what that looks like. An adaptive evolution must trace a never go down path like this one on this landscape. So along this path, we can make kind of derivative pictures like the ones that we did before to represent local topography around each point, indicating which of the possible upwards on the landscape directions is taken. So this is a picture of that. So the rule space over which our fitness landscape is defined is ultimately discrete and effectively very high dimensional, 15-dimensional for this case of k equals 2, r equals 3 halves rules. It's quite challenging to produce an interpretable version visualization of that in 3D. Well, we'd like it if we could lay out our rendering of the rule space so that rules which differ by just one mutation are a fixed kind of elementary 2D distance apart. In general, this won't be possible, but we're trying to at least approximate this by finding sort of a good layout from the underlying mutation graph. And that's what we were doing in a case like this. But using this layout here, we can in principle make a fitness landscape surface by interpolating between the discrete points that we show here. It's not completely clear how meaningful this is, but it's perhaps useful in kind of engaging our spatial intuition to be able to see things like this. We can also try machine learning and dimension reduction operating on a set of rule vectors, i.e. outcome lists, that won't be rejected in our adaptive evolution process, and the results are, well, perhaps slightly better. By the way, if we use this dimension reduction for the rule space, here's how the behavior of the rules lays out. And for comparison, here's a feature space plot just based on the visual appearance of these patterns. So the first one was based on actual mutations. The second one is simply based on visual appearance. Well, let's talk about the whole space, exhaustive search versus adaptive evolution. In adaptive evolution, we start, say, from the null rule, and then we make random mutations to try to reach rules with progressively larger fitness. But what about just exhaustively searching the complete space of possible rules? Well, the number of rules increases rapidly, becoming unmanageably large, but some cases are definitely accessible. For example, there are just 524,288 symmetric k equals 2 r equals 2 rules, of which 77,624 generate patterns with finite lifetimes. Ultimately, though, there are just 77 distinct phenotypic patterns that appear with varying lifetimes and varying multiplicity. So in this case, we're showing the multiplicity is actually, which is the numbers underneath here, are actually just directly associated with the number of unused bits that in the rule when we generate the patterns that we're showing here. So and for example, there are no unused bits in the case where there's multiplicity 1 for those patterns. So how do these exhaustive results, which we get by sampling the whole space of possible rules, compare with what's generated in the multiway graph from adaptive evolution? Well, they're almost the same in this particular case, but with 2 extra cases in the exhaustive case, which are generated by rules of the following form, where, oh, you can't see it, but there's a lot of gray cases in these rules, which are entries that don't matter. Why don't rules like this ever appear in our adaptive evolution? The reason is that there isn't a chain of point mutations starting from the null rule that can reach these rules without going through rules that will be rejected by our adaptive evolution process. If we draw a multiway graph that includes every possible acceptable rule, then we'll see a separate part in the graph with its own root that contains the rules that can't be reached by our adaptive evolution from the null rule. And here's how that works. And you can see those unreachable rules with the red connections there. So now if we look at all symmetric k equals 2 r equals 2 rules, let's look at just the distribution of lifetimes we get. Here it is. The maximum we saw above is 65. The overall distribution follows roughly a power law with exponent around -3. So what we saw above is that not all rules make use of all their bits, i.e. outcomes, in generating a particular phenotypic pattern. And what we see is that the larger the lifetime that's achieved, the more bits tend to be used. So this is the number of bits used as a function of the lifetime that's achieved. In a sense, this isn't surprising because, as we'll discuss later, we can expect to need more bits in the program to specify more elaborate behavior, or in particular, behavior that embodies a larger number for lifetime. So what about general, i.e. not necessarily symmetric, k equals 2 r equals 2 rules? There are 2 to the 31 or 2 billion of these, and if we exhaustively search through them, we find about 75 million that have finite lifetimes. And the distribution of lifetimes in that case is this. And again, it's roughly a power law, now with an exponent around -3.5. And so what do the actual patterns look like? So here are the actual 100 patterns which have the longest lifetimes. And in all the asymmetric cases here, there are also rules that giving left-right flipped patterns. So the numbers underneath in each case here are the lifetime achieved with that particular pattern. And it's really interesting to see the variety of qualitatively different ideas being used by different rules. Some of them, like, let's see, this one, like that one there, we might somehow imagine could have been constructed specifically for the purpose of having their particular long lifetimes. But other ones like this long one here with lifetime 308, somehow seem more coincidental, behaving in an apparently random way and then just happening to die out after a certain number of steps. Well, since we found these rules by exhaustive search, we know they're the only possible ones with such long lifetimes, at least with k equals 2, r equals 2. So then we can infer that the ornate structures we see are in some sense necessary to achieve the objective of, say, having a finite lifetime of more than 100 steps. So that means that if we go through a process of adaptive evolution and achieve a lifetime above 100 steps, we see a complex pattern of behavior, not because of complicated choices in our process of adaptive evolution, but rather because to achieve such a lifetime, one has no choice but to use such a complex pattern. Or in other words, the complexity we see is a reflection of computational necessity, not historical accidents of adaptive evolution. So note also that, as we'll talk about more detail later, there are certain behaviors we can get and others that we cannot. So for example, there's a rule that gives lifetime 308, but none that gives lifetime 300. Though if we use more complicated initial conditions or a more complicated family of rules, we absolutely could find such a rule. So much as we saw in the symmetric k equals 2, r equals 2 case, almost any long lifetimes require using all the available bits in the rule. And this is showing, as a function of lifetime, the number of bits, should have been labeled lifetime here, the number of bits available, number of available bits, how many of the available bits in the rule have been used. And mostly, as we've got long lifetimes, all 32 bits in the rule have been used. But needless to say, as is always the case with these adventures in the computational universe, there's an exception. A pair of rules with lifetime 84, where the outcome for the all-black 5-black cell case doesn't matter. And this is the particular rule case there. And the first cell here is supposed to be gray, indicating that it doesn't matter what its value is. These cells, remember, represent the outcomes for the succession of possible 5-cell neighborhoods in the cellular automaton. But okay, so can these long lifetime rules be reached by single mutation adaptive evolution from the null rule? Rather than trying to construct the whole multiway graph for the general k equals 2, r equals 2 case, which might have billions of nodes, starting from the null rule, we can instead construct what's in effect an inverse multiway graph, in which we start from a given rule, then successively make all single-point mutations that reach rules with progressively shorter lifetimes. And that's the result of doing that. What we see is, at least in this case, this procedure never reaches the null rule. The furthest it gets is to lifetime 2 rules. And among those rules, the closest to the null rule are these rules here. But it turns out there's no way to reach these 2-bit rules by a single-point mutation from any of the 26 1-bit rules that aren't rejected by our adaptive evolution process. In fact, this isn't just an issue for this particular long lifetime rule. It's something quite general among k equals 2 rules. And indeed, constructing the sort of the forward multiway graph, starting from the null rule, we find we can only ever reach lifetime 1 rules. So ultimately, there's a particular feature of rules with just 2 colors. It's specific to starting with something like the null rule that has lifetime 1, but it's an illustration of the fact that there can be even large swaths of rule space that can't be reached by adaptive evolution with point mutations. Well, what about symmetric k equals 2, r equals 2 rules? Well, to maintain symmetry, we have to deal with mutations that change not just 1, but 2 bits. And this turns out to mean that, except in the cases we talked about before, the inverse multiway system, starting from long lifetimes rules, always successfully reaches the null rule. And here's an example. So there's something else to notice here. Looking at this graph, we see that there's a way to get just one 2-bit mutation with just one 2-bit mutation from a lifetime 1 to a lifetime 65 rule. And that's the line over on the left, starting from the length 65 pattern at the top. And this is the mutation that's involved. And we're going, that first rule generates the lives one step phenotype that we showed on the left here. And the second rule generates the live 65 steps phenotype on the right. We didn't see this in the multiway graph that we made above because we'd done this process of transitive reduction to the graph. So we had gotten rid of these kind of direct paths rather than the paths that have to go through all the intermediate steps. If we didn't do that, we find that a few large lifetime jumps are possible. And we can see that if we plot the possible lifetimes before and after a single point mutation. So the cases where it's the same lifetime after as before would be on the line x equals y here, and the ones we can see that there are some which have a short lifetime before, but so a small x value and jump to high y value and show up at high y value, and those correspond to cases where there was a short lifetime before the mutation and a very long lifetime after the mutation. So if we go beyond k equals 2, r equals 2 rules, we can consider symmetric k equals 3, r equals 1 rules, of which there are 3 to the 17, or about 129 million. And the distribution of lifetimes in this case is like this. And again, there's roughly a power law, again with exponent around minus 3.5. And now the maximum lifetime we find is not 308, which is what we got for the symmetric k equals 2, r equals 2 rules, but instead 2,194. And here are some examples of the long-lived rules of this type. And let's see, we've got even longer ones here. And the champion here is the one on the right, 2,194 steps that it lives before dying out. So once again, there are different ideas on display here, with a few curious examples of convergence, like we can see in this case here. There are actually two rules with lifetimes 989 and 990 on the bottom row there, as well as 1,068 and 1,069, which give essentially the same patterns, but if you look carefully, they've exchanged colors, and they have one prefatory step at the very beginning that's different, but otherwise these have sort of converged to the same phenotype. So what about general, not symmetrical, k equals 3, 3 color, r equals 1, nearest neighbor rules? There are too many of these to search exhaustively, but if we do kind of directed random sampling, we get lots of long lifetime examples. Let me show you some of those. Here are some of those. We see, once again, these very different ideas on display about sort of how to live a long time. Now, the tail of very long lifetimes, in this case, extends even further. Here are examples of what happens in that case. And it's a little easier to see the kind of champion here is a lifetime 10,863 step rule. It's a little easier to see what happens if we just visualize that in sections. And we can, if we just sample 100 steps out of every 2,000 as well as piece at the end, we see sort of an elaborate alternation in this rule between periodic and seemingly random behavior. But none of it gives any obvious clue of the remarkable fact that after 10,863 steps, the whole pattern will die out. And we see its very end down there on the bottom right. The issue of undecidability. So as our example criterion for the fitness of cellular automaton rules, we've used the lifetimes of the patterns they generate, always assuming that if the patterns don't terminate at all, they should be considered to have fitness 0. But how can we tell if a pattern is going to terminate? In what we saw before, we saw patterns that live a very long time, but do eventually terminate. So here are examples of the first 100 steps of a few k equals 3, r equals 1 symmetric rules. And what will happen with these patterns? We know from what we see here that none of them have lifetimes less than 100 steps. But what would allow us to say more? In a few cases, we can see that the patterns are periodic or have obvious repeating structures, which means they'll never terminate. But in other cases, there's no obvious way to predict what will happen. Explicitly running the rules for another 100 steps, we discover some more outcomes. Going to 500 steps, though, there are some surprises. The first rule, Rule A here, becomes periodic after 388 steps. Other rules here on the bottom row terminate after respectively 265 and 377 steps. We kind of didn't see that coming from the behavior we saw above. But the question is there a way to systematically say what will happen in the end with all the remaining rules? The answer is that in general, there's not. It's not something, it's something that must be considered undecidable by any finite computation. So given how comparatively simple the cellular automaton rules we're considering are, we might have assumed that with all our sort of sophisticated mathematical and computational methods, we'd always be able to jump ahead of them and figure out their outcome without the computational effort of explicitly running each step. But the principle of computational equivalence suggests that pretty much whenever the behavior of these rules isn't obviously simple, it will in effect be of equal computational sophistication to any other system, and in particular to any methods that we might use to predict it. And the result is the phenomenon of computational irreducibility that I talk about a lot, that implies that in many systems, presumably including most of the cellular automata here, there isn't any way to figure out their outcome much more efficiently than by explicitly tracing each of their steps. So this means that to know what will happen in the end after an infinite number of steps might take an unlimited amount of computational effort. Or in other words, it must be considered effectively undecidable by any finite computation. So as a practical matter, we might look at the observed distribution of lifetimes for a particular type of cellular automaton and become pretty confident that there won't be longer finite lifetimes for that type of cellular automaton. But for the k equals 3, r equals 1 rules we talked about before, we might have been fairly confident that a few thousand steps was the longest lifetime that would ever occur until we discovered the 10,863 step example. So let's say we run a particular rule for 10,000 steps and hasn't died out. How can we tell if it never will? Well, we have to construct a proof of some kind. And that's easy to do if we can see that the pattern becomes, say, completely periodic. But in general, computational irreducibility implies that we won't be able to do it. Might there, though, still be special cases where we can? In effect, those would have to correspond to pockets of computational reusability where we can manage to find a compressed description of the cellular automaton behavior. There are cases like this, where there isn't strict periodicity, but where in the end there's basically repetitive behavior. This is a particular rule which has effective period of 480. And there are cases of nested behavior, which is never periodic, but which is nevertheless simple enough to be predictable. But there are always surprises, like this example here, that eventually resolves to have period 6, but only after 7,129 steps. So what does all this mean for our adaptive evolution process? It implies that in principle, we could miss a very long lifetime for a particular rule, assuming it to be infinite. In a biological analogy, we might have a genome that leads to unbounded, perhaps tumor-like growth, but where actually the growth in the end unexpectedly stops. Computation theoretic perspectives and busy beavers. What we're asking about the dying out of patterns in cellular automata is directly analogous to the classic halting problem for Turing machines or the termination problem for term rewriting, post-DAG systems, and so on. And in looking for cellular automata that have the longest lived patterns, we're studying a cellular automaton analog of the so-called busy beaver problem for Turing machines. We can summarize the results that we've got so far for our cellular automata. Here, all for single cell initial conditions. And if we look at kind of the profiles, the widths of non-zero cells for the patterns generated by these winning rules, here's the results that we get. And essentially, the integrals of these curves are what we've listed as what we listed as areas back here. Well, we can, for reasons we were talking about just now, we can only be certain that we found lower bounds on the actual maximum lifetime, though, except in the last few cases we looked at here, it seems very likely that we do, in fact, have the maximum lifetime. It's somewhat sobering, though, to compare with known results for maximum busy beaver lifetimes for Turing machines. Let me show you those results here, where now S is the number of Turing machine states, and the Turing machines are started from blank tapes, and they're taken to halt when they reach a particular halt state. Sufficiently small Turing machines can have only modest lifetimes, but even slightly bigger Turing machines can have vastly larger lifetimes. And in fact, as a consequence of the undecidability of the halting problem for Turing machines, that the maximum lifetime grows with the size of the Turing machine faster than any computable function, i.e. any function that can be computed in finite time by a Turing machine, or whose values can be proved by, or whose value can be proved by a finite proof in a finite axiom system. So you see some very big lifetimes there, like 10 to the 10 to the 10 to the 10, 15 times, and so on. But okay, the maximum lifetime increases with the size of the rule for a Turing machine or a cellular automaton. But what defines the size of a rule? Presumably, it should be roughly the number of independent bits needed to specify the rule, which we can think of as an approximate measure of its information content, or something like log to the base 2 of the number of possible rules of its type. So at the outset, we might imagine that all, for example, 2 to the 32, k equals 2, r equals 2 rules would need 32 bits to specify them. But as we already talked about, in some cases, some of the bits in the rule don't matter when it comes to determining the patterns they produce. And what we see is that the more bits that matter, and so have to be specified, the longer lifetimes are possible. We saw kind of a version of this picture before. Well, so far we've been discussing just cellular automata with single cell initial conditions. If we use more complicated initial conditions, what we're effectively doing is adding more information content into the system, with the result that the maximum lifetimes can potentially get larger. And as an example, here are the possible maximum lifetimes for k equals 2, r equals 3 halves rules with a sequence of possible initial conditions. As the initial conditions kind of get larger, the lifetimes get progressively slightly larger. Probabilistic approximations. So cellular automata are at their core deterministic systems. Given a particular cellular automaton rule and a particular initial condition, every aspect of the behavior that is generated is completely determined. But is there any way that we can approximate this behavior by some probabilistic model? Or might we at least usefully be able to use such a model if we look at the aggregate properties of large numbers of different rules? One hint along these lines comes from the power law distributions that we found above for the frequencies of different possible lifetimes for cellular automata of given types. And we might wonder whether such distributions, and perhaps even their exponents, like we found -3, -3.5, could be found from some probabilistic model. One possible approach is to approximate a cellular automaton by a probabilistic process, say one in which a particular cell becomes black with probability P if either it or its neighbors were black on the step before. So these are a few examples of what can happen with this what's called directed percolation setup. So the behavior varies greatly with the probability P. For small p, everything dies out, while for larger p, it fills in. And if we look at the final density, starting from random initial conditions, there's a sharp phase transition around p equals 0.54. If we instead start not from random initial conditions, but from a single black initial cell, one sees a slightly different transition. One can also plot the probabilities for different survival times or lifetimes for this pattern. And right around the transition, the distribution of lifetimes follows a power law. Actually, it's roughly -1 power law, which happens to be what one gets from the mean field theory estimate in this case. So how does this relate to cellular automata? Let's say we have a k=2 rule, and we suppose that the colors of cells can be approximated as somehow random. Then we might suppose that the patterns we get could be like in our probabilistic model, and a potential source for the value of p to use will be the fraction of cases in the rule that give a black cell as output. So if we plot the lifetimes of k=2, r=2 rules against these fractions, we see here that the longest lifetimes do occur when a little under half the outcomes are black. Though notice this is also where the binomial distribution implies the largest number of rules are concentrated. Well, if we don't try thinking about the details of cellular automata and evolution, but instead just consider the boundaries of finite lifetime patterns that we generate, we can imagine approximating these, say for symmetric rules, just by random walks, that when they collide, correspond to the pattern dying out. Well, then it turns out the standard theory of random walks tells us that the probability to survive for tau steps is approximately proportional to tau to the -3/2 for large tau. It's a power law, though not immediately one of the same ones that we've observed for our cellular automaton lifetimes. Other adaptive evolution strategies. So what we've done so far, we've always taken each step of our process of adaptive evolution to pick an outcome of equal or greater fitness. What if we adopt a more impatient procedure in which at each step we insist on an outcome that has strictly greater fitness? For k=2, it's simply not possible with this procedure, at least with a null initial condition, to escape the null rule. Everything that can be reached with one mutation still has lifetime 1. With k=3, it's possible to go one step, not very excitingly, captured by this picture here. But if we're assuming that we have to reach greater fitness with just one mutation, but what if we allow 2 mutations at a time? Well, then we can make progress. Here's the multiway system, in this case for k=2, r=2 rules, showing the number of mutations that we're making on each connection here. I should have said that. Well, we don't reach as many phenotypic patterns as by using single mutations and allowing fitness neutral moves, but where we do we get much quicker without any back and forth in fitness neutral spaces. If we allow up to 3 mutations, we get still further. And indeed, we seem to get a pretty good representative sampling of what's out there in this rule space, even though we reach only 37 rules compared to the 77,624, albeit with many duplicated phenotypic patterns from our standard approach allowing neutral moves. For k=3, r=1 symmetric rules, single mutations can only get 2 steps. But if we allow up to 2 mutations, we can go much further. And the fact that we don't now have to deal with neutral moves means we can explicitly construct at least the first few steps of the multiway graph in this case. We can go further if at each step we just pick a random higher fitness rule reached with 2 or fewer mutations. Here's an example of how that works. The adaptive evolution histories here can be generated in effect by randomly trying a series of possibilities at each step, then picking the first one that exhibits increased fitness. Another approach is to use what amounts to local exhaustive search. At each step, look at results from all possible mutations and pick one that gives the largest fitness. At least in smaller rule spaces, it's common that there will be several results with the same fitness. And as an example, we can just pick at random between these. So one might think that this approach would in fact always be an optimization of the adaptive evolution process, but in practice, the systematic character of looking around at all possible mutations can end up making it get stuck, in a sense, repeatedly just trying to do the same thing, even if it isn't working. So something of an opposite approach involves loosening our criteria for which paths can be chosen, and for example, allowing paths that temporarily reduce fitness say, by 1 step of lifetime. So those are examples of such paths. So in effect here, we're allowing less than maximally fit organisms to survive. And we can represent the overall structure of what's happening by a multiway graph, which now includes backtracking to lower fitnesses. But although the details are different, in the end, it doesn't seem as if allowing this kind of backtracking has any dramatic effect. Somehow the basic phenomena around the process of adaptive evolution are strong enough that most of the details of how the adaptive evolution is done don't ultimately matter much. An aside, sexual reproduction. So in everything we've done so far, we've been making mutations only to individual rules. But there's another mechanism that exists in many biological organisms, sexual reproduction, in which, in effect, a pair of rules, i.e. genomes, get mixed to produce a new rule. As a simple model of the crossover that typically happens with actual genomes, we can take 2 rules and splice together the beginning of one rule with the end of another, like this. In general, there'll be many ways to combine pairs of rules like this. In a direct analogy to our physics project, we can represent such recombinations as events that take 2 rules and produce 1. So the analog of our multiway graph for all possible paths of adaptive evolution by mutations is now what we call in our physics project a token event graph. So in dealing with mutations, we were able to take a single rule and progressively modify it. Now we always have to work with a population of rules, combining them 2 at a time to generate new rules. We can represent the possible combinations among one set of rules in a picture like this. And there are at this point many different choices that we could make about how to set up our model. The particular approach we'll use takes just n of the n choose 2, or n times n minus 1 over 2 possible combinations. So in this case, we might take just these possible combinations. Then for each of these selected combinations, we attempt a crossover, keeping those children, which we can draw here between their parents, that are not rejected as a result of having lower fitness. Finally, to maintain our gene pool, we can carry forward parents selected at random so that we still end up with n rules in all cases. And yes, even though we've attempted to make this whole procedure as clean as possible, it's still a mess, which seems to be inevitable and which has, as we'll discuss later, bedeviled essentially computational studies of evolution in the past all the time. Okay, so what happens when we apply this procedure, this sexual reproduction procedure, say to k equals 3, r equals 1 rules? We'll pick 4 rules at random as our initial population. And yes, 2 happen to produce the same pattern here. Then in a sequence of steps, we'll successively pick various combinations of these rules. And here are the distinct phenotype patterns produced in this process. So note that even though there can be multiple copies of the same phenotype pattern, the underlying genotype patterns are always distinct. Well, as a final form of summarization of what's going on here, we can just plot the successive fitnesses of the patterns we generate with the size of each dot reflecting the number of times a particular fitness occurs. So this is the number of rounds of evolution along the bottom, and up on the vertical axis is the fitness. We probably should label these. So in this case, we reach a steady state after 9 steps. The larger the population, the longer the adaptive evolution will typically keep going. So here are some examples with population 10 showing sort of all the patterns that are achieved. And if we just show in each case the longest lifetime rule we get so far, here's what we see in those 2 different examples. The results aren't obviously different from what we were finding with mutation above, even though now we've got a much more complicated model with a whole population of rules rather than a single rule. One obvious difference, though, is that here we can end up with overall cycles of populations of rules, whereas in the pure mutation case, that can only happen among fitness-neutral rules. So here are a few other examples, now obtained after 500 steps with population 25, and with population 50. And as far as one can tell, even here, there are no substantial differences from what we saw with mutation alone. Certainly, there are detailed features introduced by sexual reproduction and crossover, but for our purposes in understanding the big picture of what's happening in adaptive evolution, it seems sufficient to do what we've done so far and consider only mutation. An even more minimal model. So by investigating adaptive evolution in cellular automata, we're already making dramatic simplifications relative, say, to actual biology. But in the effort to understand the essence of phenomena we see, it's helpful to go even further. And instead of thinking about computational rules and their behavior, just think about vertices on a mutation graph, each assigned a certain fitness. So as an example, we can set up a 2D grid, assigning each point a certain random fitness. And then starting from a minimum fitness point, we can follow the same kind of adaptive evolution procedure as above at each point going to a neighboring point with an equal or greater fitness. Typically, we don't manage to go far before we get stuck. Though with the uniform distribution of fitness values that we're using here, we still usually end up on a fairly large fitness value. We can summarize the possible paths that one can take here by a multiway graph again. And I don't know why all these nodes are the same size. They should be different sizes in this graph. So in our cellular automaton rule space, and for that matter in biology, neighboring points don't just have independent random fitnesses. Instead, the fitnesses are determined by a definite computational procedure. So as a simple approximation, we can just take the fitness of each point to be a particular function of its graph coordinates. If the function forms something like a uniform hill, then the adaptive evolution procedure will just climb it. But as soon as the function has systematic bumpiness, there's a tremendous tendency to quickly get stuck. And if there's some unexpected spot of high fitness, adaptive evolution typically won't find it. And it certainly won't if it's surrounded by a lower fitness moat. So what happens if we increase the dimensionality of the mutation space in which we're operating? Basically, it becomes easier to find a path that increases fitness. And we can see this whole phenomenon if we look at Boolean hypercubes in increasing numbers of dimensions. But ultimately, this relies on the fact that in the neighborhood reachable by mutations from a given point, there'll be a sufficiently random collection of fitness values that it'll likely be possible to find a direction that's going up in fitness. Yet this alone won't in general be enough, because we also need it to be the case that there's enough regularity in the fitness landscape that we can systematically navigate it to find its maximum, and that the maximum is not somehow unexpected and isolated. What can adaptive evolution achieve? We've seen that adaptive evolution can be surprisingly successful at finding cellular automata that produce patterns with long but finite lifetimes. But what about other types of traits? What can and cannot adaptive evolution ultimately manage to do? For example, what if we're trying to find cellular automata whose patterns don't just live as long as possible, but instead die after a specific number of steps? It's clear that within any finite set of rules, say with particular K and R, there'll only be a limited collection of possible lifetimes. For symmetric K equals 2, R equals 2 rules, the possible lifetimes are basically these ones here. But as soon as we're dealing with even K equals 3, R equals 1 symmetric rules, it's already in principle possible, for example, to get every lifetime up to 100. But what about adaptive evolution? How well does it do at reaching rules with all those lifetimes? Let's say we do single point mutation as before, but now we accept as a mutation if it leads not specifically to a larger finite lifetime, but to a lifetime that is closer in absolute magnitude to some desired lifetime. Strictly and importantly, in both cases, we allow fitness-neutral mutations that leave the lifetime the same. So here are examples if we try to get lifetime exactly 50 in K equals 3, R equals 1 rules. It gets close. Sometimes it overshoots, but at least in these particular examples, it never quite makes it. Here's what we see if we look at the lifetimes achieved with 100 random different sequences of mutations. basically they mostly get stuck at lifetimes close to 50, but not exactly 50. It's not that k equals 3 r equals 1 rules can't yield lifetime 50. Exhaustive search shows that even many symmetric such rules can. Here are examples. It's just that our adaptive evolution process usually gets stuck before it reaches rules like these. Even though there's usually enough room to maneuver in k equals 3 r equals 1 space to get to generally longer lifetimes, there's not enough to specifically get to lifetime 50. What about k equals 4 r equals 1 rule space? There are now not 10 to the 12, but about 10 to the 38 possible rules. And in this rule space, it becomes quite routine to be able to reach lifetime 50 through adaptive evolution. And here are some examples of that. No, that's mysterious. That doesn't seem to be the right picture. Let's see. Oh yeah, there are a bunch of examples where we reach lifetime 50 directly from with 4-color rules by adaptive evolution. It can sometimes take a while, but most of the time in this rule space, it's possible to get to exactly lifetime 50. And we can see that here if we just look at the fitness as a function of number of steps. What happens with other lifetime goals? Even symmetric k equals 3 r equals 1 rules can achieve many lifetime values. Indeed, the first missing values are 129, 132, 139, and so on. And for example, many multiples of 50 can be achieved. Here are examples. But it becomes increasingly difficult for adaptive evolution to reach these specific goals. Increasing the size of the rule space always seems to help. So for example, with k equals 4, r equals 1, if one's aiming for lifetime 100, the actual distribution of lifetimes reached is this here, peaked very much around 100. In general, the distribution gets broader as the lifetime one seeking gets longer. And we saw before that across the whole space of, say, k equals 4, r equals 1 rules, the frequency of progressively larger lifetimes falls. Actually, that should be, we should have said k equals 3, r equals 1 rules. The frequency of progressively larger lifetimes falls roughly according to a power law. So this means that the fractional region in rule space that achieves a given lifetime gets progressively smaller, with the result that typically the paths followed by adaptive evolution are progressively more likely to get stuck before they reach that particular lifetime. Okay, what about other kinds of objectives? Say ones more related to the morphologies of patterns. As a simple example, let's consider the objective of maximizing the widths of finite lifetime patterns. We can try to achieve this by adaptive evolution, in which we reject any mutations that lead to decreased width. But by width, what I mean is the maximum horizontal extent of the pattern. And once again, this process manages to discover all sorts of mechanisms for achieving larger widths. Here, we're labeling every pattern by its height, i.e. its lifetime, and its width. Let's see, oh yeah, we've got some more high width patterns there. Well, so there are certainly structural constraints here. For example, the width can't be too large relative to the height, because if it's too large, patterns will tend to grow forever. But what if we specifically try to select for maximal pattern aspect ratio, i.e. ratio of width to height? In essentially every case we see there, the adaptive evolution has an effect. Well, in what we've seen so far, adaptive evolution has invented many different mechanisms for achieving the objective we've set. But here it turns out we essentially see the same idea being used over and over again, presumably because this is the only way to achieve our objective, given the overall structure of how the underlying rules we're using work. So what if we ask for something more specific? Let's say that the aspect ratio be as close to 3 as possible. Well, much of the time, the adaptive evolution finds a solution that is correct if trivial, but sometimes it finds other solutions, like these here. and often surprisingly elaborate and complicated ones. What about if our goal is an aspect ratio of around pi, 3.14? Turns out that adaptive evolution can still do rather well here, even just with symmetric k equals 3, r equals 1 rules. Here are examples. There are some examples. We can also ask about properties of the inside of the pattern. For example, we can ask to maximize the lengths of uniform runs of non-white cells in the center column of the pattern. And once again, adaptive evolution can successfully lead us to rules like, well, these random examples here, where this is large, where the length in the center column of non-white cells is large. So we can go on and get still more detailed, say asking about runs of particular lengths or the presence or number of particular subpatterns. And eventually, just like when we asked for too long a lifetime, we'll find that the cases we're looking for are too sparse, and adaptive evolution, at least in a given rule space, won't be able to find them, even if exhaustive search could still identify at least a few examples. But just what kinds of objectives or fitness functions can be handled, how well by adaptive evolution, operating, for example, on the raw material of cellular automata? It's an important question, an analog which is also central to the investigation of machine learning. But as of now, we don't really have the tools to address it. It's somehow reminiscent of asking what kinds of functions can be approximated how well by different methods or basis functions, but it's more complicated. Solving it, though, would tell us a lot about the reach of adaptive evolution processes, not only for biology, but also for machine learning. What it means for what's going on in biology. How do biological organisms manage to be the way they are with all their complex and seemingly clever solutions to such a wide range of challenges? Is it just natural selection that does it, or is there in effect more going on? And if natural selection does it, how does it actually manage it? From the point of view of traditional engineering, what we see in biology is often very surprising and much more complex and clever than we'd imagine ever being able to create ourselves. But is the secret of biology, in a sense, just natural selection? Well, actually, there's often an analog of natural selection going on even in engineering, as different designs get tried and only some get selected. But at least in traditional engineering, a key feature is that one always tries to come up with designs where one can foresee their consequences. But biology is different. Mutations to genomes just happen without any notion that their consequences can be foreseen. But one still might assume that when guided by natural selection, the results wouldn't be too different to what we'd get in traditional engineering. But there's a crucial piece of intuition missing here, and it has to do with how randomly chosen programs behave. We might have assumed, based on our typical experience with programs we explicitly construct for particular purposes, that at least a simple random program wouldn't ever do anything terribly interesting or complicated. But the surprising discovery I made in the early 1980s is that this isn't true. And instead, it's a ubiquitous phenomenon that in the computational universe of possible programs, one can get immense complexity, even from very simple programs. So this means that as mutation operates on a genome, it's essentially inevitable that it'll end up sampling programs that show highly complex behavior. At the outset, one might have imagined that such complexity could only be achieved by careful design and would inevitably be at best rare. But the surprising fact is that because of how things fundamentally work in the computational universe, it's instead easy to get. But what does complexity have to do with creating successful organisms? To create a successful organism that can prosper in a particular environment, there fundamentally has to be some way to get a genome that will solve the necessary problems. And this is where natural selection comes in. But the fact that it can work is something that's not at all obvious. There are really two issues. The first is whether a program, i.e. genome, even exists that will solve the necessary problems. And the second is whether such a program can be found by a thread of adaptive evolution that goes only through intermediate steps that are, mistake there, fit enough to survive. As it turns out, both of these issues are related to the same fundamental features of computation, which are also responsible for the ubiquitous appearance of complexity. given some underlying framework like cellular automata or like a basic apparatus of life, is there some rule that can be implemented in that framework that will achieve some particular computational objective? The principle of computational equivalence says that generically the answer will be yes. In effect, given almost any underlying hardware, it'll ultimately be possible to come up with software, i.e. a rule, that achieves almost any physically possible given objective, like growing an organism of at least some kind that can survive in a particular environment. But how can we actually find a rule that achieves this? In principle, we could do exhaustive search, but that will be exponentially difficult and in all but toy cases will be utterly infeasible in practice. So what about adaptive evolution? Well, that's the big question. And what we've seen here is that rather surprisingly, simple mutation and selection, i.e. the mechanisms of natural selection, very often provide a dramatic shortcut for finding rules that do what we want. So why is this? In effect, adaptive evolution is finding a path through rule space that gets to where we want to go. But the surprising part is that it's managing to do this one step at a time. It's just trying random variations, i.e. mutations, and as soon as it finds one that's not a step down in fitness, it'll take it and keep going. At the outset, it's certainly not obvious that this will work. In particular, it could be that at some point, there just won't be any way forward. All directions will lead only to lower fitness, and in effect, the adaptive evolution will get stuck. But the key observation from the experiments in our simple model here is that this typically doesn't happen. And there seem to be basically two things going on here. The first is that rule space is in effect very high dimensional. So that means that there are many directions to choose from in trying to find one that will allow one to take a step forward. But on its own, this isn't enough, because there could be correlations between these directions that would mean that if one's blocked in one direction, one would inevitably be blocked in all others. So why doesn't this happen? Well, it seems to be the result of the fundamental computational phenomenon of computational irreducibility. A traditional view based on experience with mathematical science had been that if one knew the underlying rule for a system, then this would immediately let one predict what the system would do. But what became clear from my explorations in the 1980s and 1990s is that in a computational universe, this generically isn't true. And instead, that the only way one can systematically find out what most computational systems will do is explicitly to run their rules step by step, doing in effect the same irreducible amount of computational work that they do. So if one's just presented with behavior from the system, one won't be in a position to decode it and see its simpler origins. Unless one's capable of doing as much computational work as the system itself, one will just have to consider what it's doing as more or less random. And indeed, this seems to be the root of many important phenomena, such as the second law of thermodynamics. And I suspect it's also at the root of the effectiveness of adaptive evolution, notably in biology. But what computational irreducibility implies is that around every point in rule space, there'll be a certain effective randomness to fitnesses one sees. And if there are many dimensions to rule space, that means it's overwhelmingly likely that there'll be paths to success in some directions from that point. But will the adaptive evolution find them? We've assumed that there are a series of mutations to the rule that all happening at random. But the point is that if there are n elements in the rule, then after some fraction of n mutations, we should find our success direction. If we were doing exhaustive search, we'd instead have to try about k to the n possible rules. At the outset, it might seem conceivable that the sequence of mutations could somehow cleverly probe the structure of rule space, knowing what directions would or would not be successful. But the whole point is that going from a rule, i.e. a genotype, to its behavior, i.e. phenotype, is generically a computational irreducible process. So assuming that mutations are generated in a computationally bounded way, it's inevitable that they can't break computational irreducibility, and so we'll experience the fitness landscape in rule space as effectively random. Okay, but what about achieving the characteristics an organism needs? What seems to be critical is that these characteristics are, in a sense, computationally simple. We want an organism to live long enough or be tall enough or whatever. It's not that we need the organism to perform some specific computationally irreducible task. Yes, there are all sorts of computationally irreducible processes happening in the actual development and behavior of an organism, but as far as biological evolution is concerned, all that matters is ultimately some computational simple measure of fitness. It's as if biological evolution is, in the sense of my recent observer theory, a computationally bounded observer of underlying computationally irreducible processes. And to the observer, what emerges is kind of the simple law of biological evolution and the idea that, yes, it is possible just by natural selection to successfully generate all sorts of characteristics. There are all sorts of consequences of this for thinking about biology. For example, in thinking about where complexity in biology comes from, is it generated by natural selection, perhaps reflecting the complicated sequence of historical accidents embodied in the particular collection of mutations that occurred, or is it from somewhere else? In the picture we've developed here, it's basically from somewhere else, because it's essentially a reflection of computational irreducibility. Having said that, we should remember that the very possibility of being able to have organisms with such a wide range of different forms and functions is a consequence of the universal computational character of their underlying setup, which in turn is closely tied to computational irreducibility. And it's in effect because natural selection is so coarse in its operation that it does not somehow avoid the ubiquitous computational irreducibility that exists in rule space, with the result that when we look inside the biological systems, we tend to see computational irreducibility and the complexity associated with it. Something that we've seen over and over again here is that, yes, adaptive evolution manages to solve a problem, but its solution looks very complex to us. There might be some simple engineering solution involving, say, a very regular pattern of behavior, but that's not what adaptive evolution finds. Instead, it finds something that to us is typically very surprising, very often an unexpectedly clever solution in which lots of pieces fit together just right in a way that our usual understand what's going on engineering practices would never let us invent. We might not have expected anything like this to emerge from the simple process of adaptive evolution, but as the models we've here studied highlight, it seems to be an inevitable formal consequence of core features of computational systems. And as soon as we recognize that biological systems can be viewed as computational, then it also becomes something inevitable for them and something we can view as in a sense, formally derivable for them. At the outset, we might not have been able to say what matters in the emergence of complexity in biology, but from the models we've studied and the arguments we've made, we seem to have quite firmly established that it's a fundamentally computational phenomenon that relies on certain general computational features of biological systems and doesn't depend on their particular detailed components and structure. But in the end, how generic is the complexity that comes out of adaptive evolution? In other words, if we were to pick programs, say, completely at random, how different would the complexity they produce be from the complexity we see in programs that have been adaptively evolved for a purpose? The answer isn't clear, though to know it would provide important foundational input for theoretical biology. One has the general impression that computational irreducibility is a strong enough phenomenon that it's the dominant force that determines behavior and produces complexity. But there's still usually something a bit different about the patterns we see from rules we found by adaptive evolution compared to rules we pick at random. Often there seems to be a certain level of apparent mechanism. The details look complicated, in some ways quite random, but there seems to be a kind of overall orchestration to what's going on. And whenever we can identify such regularities, it's a sign of some kind of computational reducibility. There's still plenty of computational irreducibility at work, but high fitness rules that we find through adaptive evolution typically seem to exhibit traces of their specialness that manifests in at least some amount of computational irreducibility. Whenever we manage to come up with a narrative explanation or a natural law for something, it's a sign that we found a pocket of computational reducibility. If we say that a cellular automaton manages to live long because it generates certain robust geometric patterns, or for that matter, that an organism lives long because it proofreads its DNA, we're giving a narrative that's based on computational reducibility. And indeed, whenever we can successfully identify a mechanism in our cellular automaton behavior, we're in effect seeing computational reducibility. But what can we say about the aggregate of a whole collection of mechanisms? In a different context, I've discussed the concept of a mechanoidal phase, distinguished, say, from solids and liquids by the presence of a bulk orchestration of underlying components. It's something closely related to what I've called class 4 behavior, and it's interesting to notice that If we look, for example, at rules we found from adaptive evolution at just here, their evolution from random initial conditions, that's those same rules, mostly shows characteristic class 4 behavior. In other words, adaptive evolution is potentially bringing us to characteristically special places in rule space, perhaps suggesting that there's something characteristically special about the kinds of structures that are produced in biological systems. And if we could find a way to make general statements about that characteristic specialness, it will potentially lead us to a framework for constructing a new broad formal theory in biology. Correspondence with biological phenomena. The models we've studied are extremely simple in their basic construction. At some level, it's remarkable that without, for example, including any biophysics or biochemistry, they can get anywhere at all in capturing features of biological systems and biological evolution. In a sense, this is ultimately a reflection of the fundamentally computational character of biology and the generality of computational phenomena. But it's very striking that even the patterns of cellular automaton behavior we see look very lifelike and organic. In actual biology, even the shortest genomes are vastly longer than the tiny cellular automaton rules we've considered. But even by the time we're looking at the length 27 genomic sequences in k equals 3, r equals 1 cellular automata, there are already 3 trillion possible sequences, which seems to be enough to see many core combinatorially driven biological phenomena, biology-like phenomena. The running of a cellular automaton rule might at first seem very far away from the actual processes that create biological organisms, involving as they do things like the construction of proteins and the formation of elaborate functional and spatial structures. But there are more analogies than one might at first imagine. For example, it's common for only particular cases in the cellular automaton rule to be used in a given region of the pattern that's formed, much as particular genes are typically turned on in different tissues in biological organisms. And for example, the geometrical restriction to a simple 1D array of cells doesn't seem to matter much as soon as there's sophisticated computation going on. We still get lots of structures that are actually surprisingly reminiscent of typical patterns of biological growth. One of the defining features of biological organisms is their capability for self-reproduction. And indeed, if it wasn't for this kind of copying, there wouldn't be anything like adaptive evolution to discuss. Our models don't attempt to derive self-reproduction, they just introduce it as something built into the models. And although we've considered several variants, we basically also just building into our models the idea of mutations. And what we find is that it seems as if point mutations made one at a time are enough to capture basic features of adaptive evolution. We've also primarily considered what amounts to a single lineage, in which there's just a single rule or genome at a given step. We do mutations, and we mostly implement natural selection just by keeping only rules that lead to patterns whose fitness is no less than we had before. If we had a whole population of rules, it probably wouldn't be so significant, but in the simple setup we're using, it turns out to be important that we don't reject fitness-neutral mutations. And indeed, we've seen many examples where the system wanders around fitness-neutral regions of rule space before finally discovering some innovation that allows it to increase fitness. The way our models are set up, that wandering always involves changes in the genotype, but usually at most minor changes in the phenotype. So it's very typical to see long periods of apparent equilibrium in which the phenotype changes rather little, followed by a jump to a new fitness level and a rather different genotype. And this observation seems quite aligned with the phenomenon of punctuated equilibrium, often reported in the fossil record of actual biological evolution. Another key feature of biological organisms and biological evolution is the formation of distinct species, as well as distinct phyla and so on. And indeed, we ubiquitously see something that seems to be directly analogous in our multiway graphs of all possible paths of adaptive evolution. Typically, we see distinct branches forming based on what seem like different mechanisms for achieving fitness. No doubt in actual biology, there are all sorts of detailed phenomena related to reproductive or spatial isolation, but in our models, the core phenomenon that seems to lead to the analog of branching in the tree of life is the existence of distinctly different computational mechanisms in different parts of rule space. It's worth noting that at least with our finite rule spaces, branches can die out with no successor species appearing in the multiway graph. And indeed, looking at the actual patterns produced by rules in different parts of the multiway graph, it's easy to imagine morphologically based taxonomic classifications that will be somewhat, if not perfectly, aligned with the phylogenetic tree defined by actual rule mutations. By the way, at a morphological level, we quite often see some level of convergent evolution in our multiway graphs. In small examples, we sometimes also see actual genomic convergence, which will typically be astronomically rare in actual biological systems. One of the remarkable features of our models is that they allow quite global investigation of the overall history of adaptive evolution. In many of the simple cases we've discussed, the rule space we're using is small enough that in a comparatively modest number of mutation steps, we get to the highest fitness we can reach. But, as the examples we saw about Turing machines suggest, expanding the size of the rules we're using even just a little can be expected to be sufficient to allow us to get astronomically further. And the further we go, the more mechanisms will be invented. It's an inevitable feature of systems that involve computational irreducibility that there are new and unpredictable things that will go on showing up forever, with new pockets of computation along with new pockets of computational reducibility. So even after a few billion years and the trillion generations and 10 to the 40th organisms or so that have ever lived, there's still infinitely further for biological evolution to go, and more branches to be initiated in the tree of life, involving more and more new mechanisms. I suppose one might imagine that at some point biological organisms would reach a maximum fitness and go no further. But even in our simple model with fitness measured in terms of pattern lifetime, there'll be no upper limit on fitness. Given any particular lifetime, it's a feature of the fundamental theory of computation that there'll always be a program that will yield a larger lifetime. So one might think at some point enough is enough. The giraffe's neck is long enough, etc. But if nothing else, competition between organisms will always drive things forward. Yes, a particular lineage of organisms achieved a certain fitness, but then another lineage can come along and get to that fitness too, forcing the first lineage to go even further so as not to lose out. Of course, in our simple model, we're not explicitly accounting for interactions with other organisms or for detailed properties of the environment, as well as countless other effects. And no doubt there are many biological phenomena that depend on these effects. But the key point here is that even without explicitly accounting for any of these effects, our simple model still seems to capture many core features of biological evolution. Biological evolution, and indeed adaptive evolution in general, is, it seems, fundamentally a computational phenomenon that robustly emerges quite independent of the details of systems. In the past few years, our physics project has given strong evidence that the foundations of physics are fundamentally computational, with the core laws of physics arising as inevitable consequences of the way observers like us parse the Rulliad of all possible computational processes. And what we've seen here now suggests that there's a remarkable commonality between the foundations of physics and biology. Both are anchored in computational irreducibility, and both sample slices of computational reducibility. Physics, because that's what observers like us do to get descriptions of the world that fit in our finite minds. Biology, because that's what biological evolution does in order to achieve the course objectives set by natural selection. The intuition of physics tends to be that there are ultimately simple models for things, whereas in biology, there's a certain sense that everything is always infinitely complicated with a new effect to consider at every term. But presumably that's in large part because what we study in biology tends to quickly come face to face with computational irreducibility, whereas in physics, we've been able to find things to study that avoid this. But now the commonality and foundations between physics and biology suggests that there should also be in biology the kind of structure we have in physics, complete with general laws that allow us to make useful, broad statements. And perhaps the simple model I'm talking about here can help lead us there, and in the end, help us build a new paradigm for thinking about biology in a fundamentally theoretical way. I had some historical notes that I'll go through as well. So there's a long, if circuitous, history to the things I've talked about here. Basic notions of heredity, particularly for humans, were already widely recognized in antiquity. Plant breeding was practiced from the earliest days of agriculture, but it wasn't until the late 1700s that any kind of systematic selective breeding of animals began to be commonplace. Then in 1859, Charles Darwin described the idea of natural selection, whereby competition of organisms in their natural environment could act like artificial selection, and he posited would over long periods of time lead to the development of new species. He ended his big book, Origin of Species, with the following claim, that from the war of nature, the production of the higher animals directly follows, and whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. So what he appears to have thought is that there would somehow follow from natural selection a general law, like the law of gravity, that would lead to the evolution of progressively more complex organisms, culminating in what he called the higher animals. But absent the kind of model I'm discussing here, nothing in the later development of traditional evolutionary theory really successfully supported this, or was able to give much analysis of it. Right around the same time as Darwin's Origin of Species, Gregor Mendel began to identify simple probabilistic laws of inheritance. And when his work was rediscovered at the beginning of the 1900s, it was used to develop mathematical models of the frequencies of genetic traits in populations of organisms, with key contributions to what became the field of population genetics being made in the 1920s and 1930s by J.B.S. Haldane, R.A. Fisher, Sewell Wright, and so on, who came up with, Sewell Wright came up with the concept of a fitness landscape. On a quite separate track, there have been efforts ever since antiquity to classify and understand the growth and form of biological organisms, sometimes by analogy to physical or mathematical ideas. And by the 1930s, it seemed fairly clear that chemical messengers were somehow involved in the control of growth processes. But the mathematical methods used in population genetics basically only handled discrete traits, or simple numerical ones accessible to biometry, and didn't really have anything to say about something like the development of complexity in the forms of biological organisms. The 1940s saw the introduction of what amounted to electrical engineering-inspired approaches to biology, often under the banner of cybernetics. Idealized neural nets were introduced by Warren McCulloch and Walter Pitts in 1943, and soon the idea emerged, notably in the work of Donald Hebb in 1949, that learning in such systems could occur through a kind of adaptive evolution process. And by the time practical electronic computing began to emerge in the 1950s, there was a widespread belief that ideas from biology, including evolution, would be useful as an inspiration for what could be done. Often, what now would just be described as adaptive algorithms were couched in biological evolution terms, and even when iterative methods were used for optimization, say in industrial production or engineering design, they were sometimes presented as being grounded in biological evolution. Meanwhile, by the 1960s, there began to be what amounted to Monte Carlo simulations of population genetics-style evolutionary processes. A particularly elaborate example was work by Niels Barcelli on what he called numeric evolution, in which a fairly complicated numerical cellular automaton-like competition between organisms program with randomness injected from details of data layout and computer memory showed what he claimed to be biological evolution-like phenomena, such as symbiosis and parasitism. In a different direction, there was an attempt, notably by John von Neumann, to mathematicize the foundations of biology, leading by the late 1950s to what we now call two-dimensional cellular automata, engineered in complicated ways to show phenomena like self-reproduction. The follow-up to this was mostly early theoretical computer science work with no particular connection to biology and no serious mention of adaptive evolution. When the game of life was introduced in 1970, it was widely noted as doing lifelike things, but essentially no scientific work was done in this direction. By the 1970s, though, L systems and fractals had introduced the idea of recursive tree-like or nested structures that could be generated by simple algorithms and rendered by computer graphics, and seemed to give forms close to some seen in biology. My own work in one-dimensional cellular automata, starting in 1981, focused on systematic scientific investigation of simple programs and what they do, with the surprising conclusion that even very simple programs can produce highly complex behavior. But while I saw this as informing the generation of complexity in things like the growth of biological organisms, I didn't at the time, as I'll talk about a bit later, end up seriously exploring any adaptive evolution angles. Still another thread of development concerning applying biological-like evolution not just to parameters, but to operations in programs was followed. So, for example, in 1958, Richard Friedberg at IBM tried making random changes to instructions in machine code programs, but didn't manage to get this to do much. 20 years later, super optimizers and practical compilers did begin to successfully use such techniques. Then in the 1960s, John Holland, who had at first studied learning in neural nets and was then influenced by Art Burks, who'd worked on cellular automata with von Neumann, John Holland suggested representing what amounted to programs by simple strings of symbols that could readily be modified like genomic sequences. The typical idea was to interpret the symbols as computational operations and to assign a fitness based on the outcome of these operations. A genetic algorithm could then be set up by having a population of strings that was adaptively evolved. Through the 1970s and 1980s, occasional practical successes were reported with this approach, particularly in optimization and data classification, with much being made of the importance of sexual reproduction-inspired crossover operations. Something that began to be used in the 1980s was also the much simpler approach of simulated annealing that involves randomly changing values rather than programs. By the beginning of the 1980s, the idea had also emerged of adaptively modifying the structure of mathematical expressions and of symbolic expressions representing programs. There were notable applications in computer graphics, for example, by Carl Sims, as well as to things like the 1984 Core War game involving competition between programs in a virtual machine. In the 1990s, John Koza was instrumental in developing the idea of genetic programming, notably as a way to automatically create inventions, for example, in areas like circuit and antenna design. And indeed, to this day, scattered applications of these methods continue to pop up, particularly in geometrical and mechanical design. From the very beginning, there had been controversy around Darwin's ideas about evolution. First, there was the issue of conflict with religious accounts of creation, but there were also often vigorous disagreements within the scientific community about the interpretation of the fossil record and about how large-scale evolution was really supposed to operate. A notable issue, still very active in the 1980s, was the relationship between the freedom of evolution and the constraints imposed by the actual dynamics of growth in organisms and interactions between organisms. And despite much insistence that the only reasonable scientific, as opposed to religious, point of view was that natural selection is all there is, there were nagging mysteries that suggested there must be other forces at work. Building on the possibilities of computer experimentation, as well as things like my own work on cellular automata, there emerged in the mid-1980s, particularly through the efforts of Chris Langton, a focus on investigating computational models of artificial life. This resulted in all sorts of simulations of ecosystems and so on that did produce a variety of evolution-related phenomena known from field biology, but typically the models were far too complex in their structure for it to be possible to extract fundamental conclusions from them. Still, there continued to be specific, simpler experiments. For example, in 1986, for his book, The Blind Watchmaker, Richard Dawkins made pictures of what he called biomorphs, produced by adaptively adjusting parameters for a simple tree growth algorithm based on the overall shapes generated. In the 1980s, stimulated by my work, there were various isolated studies of rule evolution in cellular automata, as well as art and museum exhibits based on this. In the 1990s, there was more systematic work, notably by Jim Crutchfield and Melanie Mitchell, on using genetic algorithms to try to evolve cellular automata rules to solve tasks like density classification. Around this time, evolutionary computation also began to emerge as a general term covering genetic algorithms and other usually biology-inspired adaptive computational methods. Meanwhile, accelerating in the 1990s, there was great progress in understanding actual molecular mechanisms in biology and in figuring out how genetic and developmental processes work. But even as huge amounts of data accumulated, enthusiasm for large-scale theories of biology that might, for example, address the production of complexity in biological evolution seemed to wane. The discipline of systems biology attempted to develop specific, usually mathematical models for biological systems, but there never emerged much in the way of overarching theoretical principles, except perhaps somewhat specifically in areas like immunology and neuroscience. One significant exception in terms of fundamental theory was Greg Chaitin's concept from around 2010 of metabiology, an effort I'll talk about in a little bit, to use ideas from the theory of computation to understand very general features of the evolution of programs and relate them to biological evolution. Starting in the 1950s, another thread of development, sometimes viewed as a practical branch of artificial intelligence, involved the idea of machine learning. Genetic algorithms were one of half a dozen common approaches. Another was based on artificial neural nets. For decades, machine learning languished as a somewhat esoteric field dominated by engineering solutions that would occasionally deliver specific application results. But then in 2011, there was unexpectedly dramatic progress in using neural nets for image identification, followed in subsequent years by successes in other areas, and culminating in 2022 with the arrival of large language models and ChatGPT. What hadn't been anticipated was that the behavior of neural nets can change a lot if they're given sufficiently huge amounts of training. But there isn't a good understanding of just why this is so and just how successful neural nets can be at what kinds of tasks. Ever since the 1940s, it has been recognized that there are relations between biological evolution and learning in neural nets. And having now seen the impressive things neural nets can do, it seems worthwhile to look again at what happens in biological evolution and to try to understand why it works, not least as a prelude to understanding more about neural nets and machine learning. Well, I also included some personal notes, a little bit of personal history, which people might find fun. So I have to say, it's strange to say, but most of what I've talked about here, I should really have figured out 40 years ago. And I almost did, except that I didn't try quite the right experiments and didn't have the intuition to think it was worth trying more. 40 years later, I have new intuition, particularly informed by experience with modern machine learning. But even now, what made possible what I've done here was a kind of chance experiment done for a somewhat different purpose. Back in 1981, I had become very interested in the question of how complexity arises in the natural world, and I was trying to come up with models that might capture this. Meanwhile, I just finished version 1 of SMP, the forerunner to Mathematica in the Wolfram language, and I was wondering how one might generalize its pattern matching paradigm to general AI. As it happened, right around that time, neural nets gained some temporary popularity, and seeing them as potentially relevant to both my topics, I started simulating them and trying to see what kind of general theory I could develop about them. But I found them frustrating to work with. There seemed to be too many parameters and details to get any clear conclusions, and at a practical level, I couldn't get them to do anything particularly interesting. But I decided that for my science question, I needed to come up with something much simpler. And as a kind of minimal merger of spin systems and neural networks, I ended up inventing cellular automata. Only later did I discover that versions of them had been invented several times before. As soon as I started doing experiments on cellular automata, I discovered that they were a window into an amazing new scientific world that I've continued to explore in one way or another ever since. My key methodology, at least at first, was just to enumerate the simplest possible cellular automaton rules and see what they did. The diversity and complexity of their behavior was remarkable, but the simplicity of the rules meant that the details of successive rules were usually fairly different. And while there were common themes in their overall behavior, there didn't seem to be any particular structure to rule space. Occasionally, though, particularly in finding examples for exposition, I would look at slightly more complicated and multicolored rules, and I certainly anecdotally noticed that rules with nearby rule numbers often had definite similarities in their behavior. It so happened that around that time I started publishing about cellular automata in 1983. There was a fair amount of ambient interest in theoretical biology, and perhaps in part because of the cellular in cellular automata, I was often invited to theoretical biology conferences. People would sometimes ask about adaptation in cellular automata, and I would usually just emphasize what individual cellular automata could do without any adaptation and what significance it might have for the development of organisms. But in 1985, I was going to a conference at Los Alamos on evolution, games, and learning, and I decided I should take a look at the relationship of these topics to cellular automata. But too quickly, I segued away from investigating adaptive evolution to seeing what kind of pattern matching and other operations I might be able to set cellular automata explicitly up to do. And as an example of the paper that I wrote at that time. Actually, in many aspects of this paper still seem quite modern. In fact, should probably be investigated more now. But even though I absolutely had the tools to do it, I simply failed at that time to explore what I've now explored. Back in 1984, I wrote a paper called 20 Problems in the Theory of Cellular Automata, and problem 7 was, how is different behavior distributed in the space of cellular automata rules? And over the years, I'd occasionally think about cellular automata rule space, wondering, for example, what kind of geometry it might have, and particularly in the continuum limit of infinitely large rules. By the latter half of the 1980s, theoretical biology conferences had segued to artificial life ones. And when I went to such conferences, I was often frustrated. People would show me simulations that seemed to have far too many parameters to ever be able to conclude much. People would also often claim that natural selection was a very simple theory, but as soon as it was implemented, there'll be all kinds of issues and choices to be made about population sizes, fitness cutoffs, interactions between organisms, and so on. And the end result was usually a descent into some kind of very specific simulation without obvious robust implications. You know, in the mid-1980s, I put a fair amount of effort into developing both the content and the organization of a new direction in science that I called complex systems research. My emphasis was on systems like cellular automata that had definite simple rules but highly complex behavior. Gradually, though, complexity started to be a popular buzzword, and I suspect partly to distinguish themselves from my efforts, some people started emphasizing that they weren't just studying complex systems, they were studying complex adaptive systems. But all too often, this seemed mostly to provide an excuse to dilute the clarity of what could be studied, and I was sufficiently put off that I paid very little attention. By the mid-90s, I booked A New Kind of Science, and I wanted to use biology as an example application of my methodology and discoveries in the computational universe. In a section that I called Fundamental Issues in Biology, I argued, much as I have here, that computational reducibility is a fundamentally stronger force than natural selection, and that when we see complexity in biology, it's most likely of computational origin rather than being sculpted by natural selection. And as part of that discussion, I included a picture of the often somewhat gradual changes in behavior that, show you this, that one sees with successive one-bit changes in a three-color nearest neighbor cellular automata rule. And yeah, in the version of the book was not in color, so you couldn't see the three colors here. But this wasn't done adaptively. It was basically just looking along a random straight line in rule space. And indeed, both in this page and in most of the book, I was concerned with what systems like cellular automata naturally do, not what they can be constructed or adaptively evolved to do. I did give constructions of how cellular automata can perform particular computational tasks, like generating primes, and somewhat obscurely, in a section on intelligence in the universe, I explored finding three-color nearest neighbor rules that can successfully double their input. My reason for discussing these rules was to highlight the difficulty of saying whether one of these cellular automata was constructed for a purpose or just doing what it does. That's the page of doubling cellular automata. Well, many years went by. There'd be an occasional project at our summer school about rule space and occasionally about adaptation. I maintained an interest in foundational questions in biology, gradually collecting information and sometimes giving talks about the subject. Meanwhile, though I didn't particularly internalize the connection then, by the mid-2010s, through our practical work in the Wolfram language, I'd gotten quite up to speed with modern machine learning. Around the same time, I'd also heard from my friend Greg Chaitin about his efforts, as he put it, to prove Darwin using the kind of computational ideas he'd applied in thinking about the foundations of mathematics. Then in 2020 came our physics project with its whole formalism around things like multiway graphs. It didn't take long to realize that, yes, what I was calling multi-computation wasn't relevant just for fundamental physics. It was something quite general that could be applied in many areas, which by 2021, I was trying to catalog. I did some thinking about each of these. The one I tackled most seriously first was metamathematics, about which I finished a book in 2022. Late that year, I was finishing a kind of 50-year in gestation project informed by our physics project on understanding the second law of thermodynamics. And as part of this, I made what I thought was some progress on thinking about the fundamental character of biological systems, though not their adaptive evolution. And then ChatGPT arrived. And in addition to being involved with it technologically, I started to think about the science of it, and particularly about how it could work. Part of it seemed to have to do with unrecognized regularities in human language, but part of it was a reflection of the emerging meta-discovery that somehow if you bashed a machine learning system hard enough, it seemed like it could manage to learn almost anything. Why did this work? At first, I thought it must just be an obvious consequence of high dimensionality, but I soon realized there was more to it. And as part of trying to understand the boundaries of what's possible, I ended up a couple of months ago writing a little piece exploring, can AI solve science? So I talked about different potential objectives for science, making predictions, generating narrative explanations, and so on. And deep inside the piece, I had a section entitled Exploring Spaces of Systems, in which I talked about science problems of the form, can one find a system that does X? And asked whether systems like neural nets could somehow let one jump ahead in what would otherwise be huge exhaustive searches. Well, as a sideshow to this, I thought it might be interesting to compare with what a non-neural net adaptive evolution process could do. Remembering Greg Chasen's idea about connecting the halting problem to biological evolution, I wondered if perhaps one could just adaptively evolve cellular automaton rules to find ones that generated a pattern with a particular finite lifetime. I imagined it as a classic machine learning problem with a loss function one needed to minimize. And so it was that just after 1 a.m. on February 22nd this year, I wrote 3 lines of Wolfram language code and tried an experiment. And it worked. And I managed to find cellular automaton rules that would generate patterns, in this particular case, living exactly 50 steps. Well, in retrospect, I was slightly lucky. First, that this ended up being such a simple experiment to try, at least in Wolfram language, that I did it even though I didn't really expect it to work. And second, that for my very first experiment, I picked parameters that happened to immediately work, 4 colors, lifetime 50, and so on. But yes, I could in principle have done the same experiment 40 years ago, though without the Wolfram language, it wouldn't have been so easy. Still, with the computers I had back then, they were powerful enough that I could in principle have generated the same results then as now. But without my modern experience in machine learning, I don't think I would have tried. And I certainly would have given up too easily. And yes, it's a bit humbling to realize that I've gone so many years assuming adaptive evolution was out of the reach of simple, clean experiments. But it's satisfying now to be able to check off another mystery I've long wondered about, and to think that much more about the foundations of biology and machine learning might finally be within reach. And that's the end of my piece that I posted a week or so ago. All right, I think I have time for some questions here. So let me see what people would like to talk about. Lewis asks, would it be worth investigating this where fitness is expressed in terms of entropy rather than lifetime? Some sort of proxy of entropy, such as the compressed size, the size of a compressed representation of the evolution. What if we were to measure the entropy of a sliding window over the evolution? Does the derivative tell us anything? Yeah, that's an interesting idea. One could try looking at, just look at the pattern one generates by running the cellular automaton rule and ask what its maximum compressed size is using some particular compression measure, like some kind of zip compression or something like that, or for extra points, the amount of compression that a neural net could give it. Yeah, it's an interesting idea. It's going to end up being kind of another level above lifetime, because if you say, let us a pattern run for as long as it can run for, it'll often run for an infinite time. I suppose one could do this particular experiment by saying, run for 100 steps, take the pattern you get then, see what the compressed pattern is, and use that as essentially a loss function or a fitness function to see how one would evolve. It's a good idea. You should try it. By the way, I might say that in the piece that I wrote, every picture, you can click it and you'll get the Wolfram language code to run that picture, so you can make your own versions of this. Let's see, LCQ asks, to what extent does the organism modifying its environment as strategy to optimize its own fitness and vice versa? i.e. complex mutual recursion, limit the usefulness of organism-focused CAs. Yeah, I mean, I wondered about that. I'd always assumed that the details of the environment and the whole structure of, what is the structure of the environment, how do different organisms interact with the organism one's looking at, and so on, that all of that will be crucial to being able to see what the essence of adaptive evolution actually is. To my surprise, that doesn't seem to be necessary. There's sort of a core phenomenon that has to do with kind of computational irreducibility, meeting sort of the coarseness of adaptive constraints and so on. That seems to be something one can investigate without all of that extra detail of organisms interacting with others and so on. And by the way, I mean, by looking at these multiway graphs and such like, one is seeing a window into sort of the all possible organisms of a population and what they do. Let's see, Tor asks, do you know any objective way to determine whether a given feature is a result of drift versus adaptation, or is there always some interpretation involved? It's a good question. It's a good question. I mean, whether some particular feature, some particular bit is the result of kind of fitness neutral drift, or whether it is something that was sort of put there for a purpose to achieve an objective. It's a good and fundamental question. I mean, certainly it's a good thing to do to look at the particular examples I have where you can ask, does this particular structure arise? Does this particular structure, arising and disappearing as you neutrally drift? Or does this structure only arise when you go through sort of forward evolution to achieve a better fitness, so to speak? That's a good thing to look at. And in fact, one can imagine looking at that in the specific examples that I have, where we can just ask, let's see, where's a good example? We could just ask for some of these rules. I don't know, let me put up a rule here. And we could ask whether the particular kind of triangular areas, do they sort of arise and disappear? This doesn't show the neutral steps, but do they arise and disappear in the neutral steps so that they sort of don't matter and you could have them or not? Or are they things that sort of have to arise to make forward progress? I think that's the kind of thing one could investigate. But what's nice is that one has explicit examples here. One can explore things like that. Another good question there. Krista asks, how about introducing topology to challenge the patterns of the cellular automaton so that different height requires different automata so that competition can start on a 3D domain? Well, gosh, I mean, yes, one could look at different rules kind of laid out on a grid, for example. In fact, Greg Chaitin was just suggesting that to me. And it's a good experiment to try to look at sort of local competition between rules in addition to just looking at a rule trying to achieve higher fitness. So you could say, of these two nearby rules on this grid, which one achieves higher fitness? I'm not sure how different the results are going to be from what we've already seen, because it's really a question of, are you exploring purely in rule space, or are you constrained just by steps in rule space, or are you also constrained by steps in physical space? But it's a good thing to look at. LCQ task, to what extent can algorithmic information theory be used to characterize and as a metric for the optimality of biological evolution processes over these possible spaces and paths? Well, I don't know. What we've got here is rules with a certain number of bits that matter. And in a sense, the number of bits that matter you can think of as being sort of the algorithmic information content of these rules. And what we're seeing is that with a very small number of bits that matter, we can achieve lots of fitness, so to speak. Now, Greg Chaitin has an approach that's based on machines with oracles and much more sort of hyper-computational thinking. I think it's going to be a story of our long-running discussion about whether the universe is like omega, his non-computable constant that represents the halting probability of a universal Turing machine, just had its 50th anniversary, actually, or whether the universe is like pi, something explicitly computable. I think this is going to be the same kind of story, but I haven't quite unraveled how that works. let's see, Elsie asks, what about the complex composite fitness of both organisms, the population, and the environment, where they mutually co-evolve, both defining and assessing what's going on? Yes, there are an infinite number of ways that one can make these models more complicated. The achievement that I was sort of excited about here was we got something that really seems to capture some core features of biological evolution with a model that we can kind of wrap our arms around. As soon as we start talking about co-evolution and things like this, we're dealing with something where we're going to have boatloads of parameters. We're going to have much more complicated models. So I say, I mean, I have been saying this about biological evolution in general, so it's probably worth the thought to see whether there are some similarly simple models that involve sort of multi-organism competition and so on. But I don't know that. And so far, the surprise is that just one organism, one lineage with pure mutation is sufficient to show some pretty interesting and core phenomena, I think. Is there let's see, RBS asks, there's a strict mathematical reason for using 26 dimensions. Oh gosh, it sounds like string theory, doesn't it? Or can any power of 2 number of dimensions be used? The reason that number shows up is just because there are 3 to the 3 possible cases in a 3-color rule with nearest neighbors, which is 27 cases. We're cutting off one of those cases because we want all white to turn to all white. So we keep the sort of background blank. And then and that leaves us with 26 cases in the rule. And then every mutation involves taking one of the 3 colors that you already have and going to one of the other 2 colors. So it's 2 times, let's see, it's 2 times 26 altogether, which is 52 possible directions. But I see 20, yeah, that's right. It's 26 dimensions because there are 26 changes you can make to the rule. They're discrete dimensions, and each one has 3 possible values. So there are 2 possible directions you can go away from the one you're already at, so to speak. 2 possible positions you can go away from the one you're already at. But this is just because of the particular cellular automaton rules I picked. I mean, with larger rules, you get much larger dimensional spaces and so on. Let's see. Yes, commenting on that, it's nice to see one can compare the efficiency of biological evolution, comparing it with exhaustive search. Yes. It is interesting. I was really excited about it. I am really excited about it. There's a lot more to study, but I sort of thought I would put out what I had figured out so far. LC comments that one of my presentations on a given topic will converge to a discussion to computational irreducibility is itself quite computationally reducible. I do tend to agree. One of my kids showed me some posts from somebody that noted that in a thing that I wrote about the 3-body problem and eclipse prediction, that there was a discussion of computational irreducibility there, which is highly relevant to the 3-body problem. But they described it as a kind of principle of narrative equivalence, analogous to my principle of computational equivalence, that in the end, anything I talk about ends up talking about computational irreducibility. I think the reason for that, I would argue, is because it's a really important phenomenon. And it's in a sense, it's the banner phenomenon of the computational way of thinking about the world. And so it's really something that's going to show up whenever you have a computational way of thinking about the world. LC comments that I might find it interesting that my work on Wolfram language design, whether streamed or not, can itself be the object of evolutionary analysis. Yes, indeed, a good thing to study. I have studied it a little, and I have all the data to study kind of the evolution under, well, I don't know if it's natural selection or human-imposed artificial selection of the features of Wolfram language. It would be a good thing to study at some point. Natanan asks, how have computational models influenced our understanding of evolutionary dynamics and species interactions? I mean, there's a huge amount of literature on this subject. And there are lots of models that use, for example, two-dimensional cellular automata to discuss, I mean, plants are easier than animals because they don't move around, they just sit there. And so you can say, a plant interacts with its neighbors and so on. And there are lots of cool results about sort of the banding patterns in forests and things that you get from simple cellular automaton rules. There's a lot of results of that kind. I would say that my sort of big picture view of that is a lot of results about fairly simple properties of what happens, because one hasn't really had a good way to talk about sort of the more complicated things that happen. What I'm showing here, and I don't think it's a beginning, not an end, is here are sort of complicated things that can happen, which you can see are visually complicated just from these very simple rules that are sort of driven by adaptive evolution. So I think, I mean, there's, as I say, there's a huge amount of literature on computational models. People have used a bunch of models I've developed with cellular automata. People have used Wolfram language a lot. I've been quite exposed to these things, but they're not really quite addressing these foundational questions in biology. They're more addressing questions that are more in the, we see this particular thing in this particular class of systems and so on. Let's see, Natanan asks, are there universal properties that all computationally irreducible systems share? Mostly that they are computationally irreducible, I'm afraid. That there are many, many consequences of computational irreducibility in terms of apparent randomness, lack of compressibility, kind of always generating surprises. There are all sorts of kind of implications. It's kind of like and closely related to things like the law of entropy increase, the fact that things tend to get to more random, higher entropy, then has many, many implications. So does the presence of computational irreducibility. 420 asks, what insights of simulations of artificial life provided in understanding the fundamental processes of natural evolution? Well, artificial life is sort of a great initiative. I would say that my own feeling is that people have tried to be too realistic in most of the simulations that have been done. They've tended to try and say, hey, let's simulate a whole rainforest. Let's get, 100 different species, and there are more than that in the rainforest, but, and let's study the way that this is a parasite of that, and the way this, and so on, and so on, and so on. And they have quite realistic models, in a sense, which, yes, actually do seem to reproduce what is seen in practice. But in a sense, that's not too surprising, because you really put in the effects that are really happening in practice, and it's the interaction of those effects that give the things you see. So it's something that I would say, in terms of these foundational questions about sort of why does biological evolution work, why is there complexity in biological organisms and so on, I think that's not been the focus of a lot of what's been done and what's generally referred to as artificial life. It's been more exploring the sort of more realistic examples. Now, that's probably not generally true. I mean, there are endless things that have been written, and a bunch of them, particularly in the earlier years, were very much based on cellular automata and on things that I'd explored and so on in cellular automata. But again, with a somewhat different emphasis. I mean, there were a lot of nice results, like Chris Langton had a nice result that sort of class 4 behavior was in between class 2 and class 3. If you looked at various different sequences of rules and so on. Okay, let's see. Last question here. RBS asks, I'm curious whether there's more irreducibility or more reducibility in these rules. How can we measure the density of irreducibility or reducibility? I think any of these kind of compression measures of, to what extent can this particular procedure predict what's going to happen is a way to sort of explore reducibility. And it's a good suggestion that you guys are making, should be investigated. We have our annual summer school and summer research program for high school students coming up in a few weeks, I guess. That's great fodder for some projects for students there. And I can't help but to advertise those programs because they're always a lot of fun, I think, for everybody involved. Certainly for me, I kind of invent lots and lots of projects for people, which people seem to have a lot of fun with, and I learn a lot from those people and from the projects that they do. All right, well, we should wrap up here, and thanks for coming, and I hope you find my efforts on biological evolution, which I wasn't expecting to write about at this time, but happened to kind of chance upon some interesting directions. I hope you found them interesting. I'm hoping to apply these ideas to thinking about the foundations of machine learning, and hopefully I'll have something to say about that in due course. Also, some of what I've talked about here is relevant to questions about sort of why observers like us exist. I've talked about how observers like us inevitably observe certain laws of physics, but there are questions about why observers like us exist. And I think that some of this sort of work on adaptive evolution and so on might shed some light on that, and about why there is this patch of rulal space that is us and all the things we know about, and how that manages to arise and manage to stay coherent. And perhaps the things that I've done here will shed some light on that. So anyway, thanks so much for joining me, and bye for now.